Hello students, today I will be starting with the chapter that is cell, the unit of life. I have discussed in the previous chapter, that is the living world, that one of the defining feature, one of the characteristic feature of the living organism is cellular organization. Anything less than the structure of cell does not ensure independent living. So in this chapter, I will be discussing the details of the cells, both the prokaryotic as well as the eukaryotic cells. But before starting with the details of the cellular structure, I will be dealing with the contribution of some important scientists towards the discovery of cell as well as formulation of cell theory. So let's start with it. Cell, the unit of life. So let's start first with the historical account. Now first of all, here I will be discussing the contribution of the Robert Hooke. Robert Hooke, he cut the thin slice of cork and observed this slice under his own design microscope. So he observed thin slice of cork, and in the thin slice of cork he examined this type of structure. Yes, so he observed this type of the structure in the thin slice of cork. This structure, it is honeycomb like structure. अब इस honeycomb like structure के अंदर ये जो compartments नजर आ रहे हैं, yes, these boxes which are appearing here, these boxes he called them as cellulae. Yes, these compartments he called them as cellulae. Singular is cellular, and today we know that these cellulae, they represent cells. Yes, they are known as cells. Actually, Robert Hooke ने ये जो cells observe करे, these are the dead cells. Yes, these are the dead cells. तो आपको मालूम है कि cell के अंदर जो living material present होता है, that is protoplasm. Yes, protoplasm is the living material of the cell. पर यहाँ पर dead cells हैं, तो protoplasm degenerate कर गया है, जिसकी वजह से ये empty space बनी है, right? और ये जो part यहाँ पर दिख रहा है, this part is going to represent the cell wall. तो रॉबर्ट हुक ने फाइंड करा honeycomb like structure when he observed the thin slice of cork, but the cells which were observed by the Robert Hooke, they were dead cells, only represented by the cell wall. Now, after the Robert Hooke, the discovery of living cell is credited to the Leeuwenhoek. Yes. For the first time, he observed the living cells. तो जो living cells Leeuwenhoek ने observe करे, that is bacteria, protozoa, spermatozoa, and RBC, it is very well known to you, RBC, that is the red blood cells. Now the nucleus inside the cell, it was first discovered by the scientist Robert Brown. Yes. So the Robert Brown, he discovered the nucleus in the cell, and he discovered it from the orchid root cells. So it is very well known to you that the nucleus, it contains genetic material, it contains hereditary material, and the nucleus control all the activities occurring inside the cell. Now the another one is Fontana. Fontana discovered nucleolus inside the nucleus. Yes, nucleolus, this is part of the nucleus, and this nucleolus, it is involved in the formation of special type of the RNA that is known as ribosomal RNA. तो nucleolus discover करा Fontana ने, and this was observed in the skin cells of eel. Now, after the discovery of the electron microscope, now the detailed structure of the cell was discovered. Now, after this, now we can see some of the contribution associated with the two scientists, that is the Schleiden and the another one is Schwann. Schleiden, he was a German botanist. And in the year 1838, what he stated that all the plants, they are composed of one or more than one cells. So he examined several plant tissue. After examining these plant tissues, what he stated that all plants, they are made up of one or more than one cells. Similarly, coming to the Schwann in 1839, he was a German zoologist. He observed several animal tissues. After examination of the several animal tissues, he stated that all animals, they are composed of cells. They are formed of cells. In the cell, there is presence of the nucleus, and the cell is enclosed by a thin membrane, that is the cell membrane. The cell is enclosed by cell membrane, but in the case of the animal cells, the cell wall is absent. So plant cell के आसपास thick covering, thick layer cell wall present है, but in the case of the animal cell, this thick covering is absent. Only there is presence of thin cell membrane. Now, Schleiden और Schwann ने जब ये state करा कि all plants, they are made up of these cells, all animals, they are made up of these cells. So this is how they formulated the cell theory. So now the cell theory which was given by the Schleiden and Schwann, I will be discussing some of the important points associated with it. So let's study some of the important points associated with the cell theory. So the cell theory was given by the two scientists, Schleiden and Schwann. Now, according to the cell theory, all living organisms, they are composed of cells and their products. तो जितने भी living organisms हैं, they are made up of cells. So we can simply say that the cell represents structural unit of all living organisms, right? So all living organisms are composed of cells. And their products. So on this basis, we can easily say that the cell represents structural unit of all living organisms. Now each cell, it consists of mass of protoplasm. Yes, तो हर एक सेल के अंदर देखो क्या प्रेजेंट है, हर एक सेल के अंदर प्रेजेंट है मास ऑफ प्रोटोप्लाज्म. नाउ व्हाट इज दिस मास ऑफ द प्रोटोप्लाज्म? प्रोटोप्लाज्म के अंदर आएगा साइटोप्लाज्म. Yes, this is cytoplasm and it is enclosed by the cell membrane. Yes, this is enclosed by the cell membrane. Cytoplasm and the membrane together, this is going to represent प्रोटोप्लाज्म. और अगर ये प्लांट सेल है तो सेल वॉल प्रेजेंट होगी अराउंड द सेल मेम्ब्रेन, और अगर ये एनिमल सेल है देन द सेल वॉल इज एब्सेंट. सो अकॉर्डिंग टू दिस थ्योरी, ईच सेल कंसिस्ट ऑफ मास ऑफ प्रोटोप्लाज्म, that is cytoplasm plus plasma membrane. और अगर प्लांट सेल होगा तो सेल वॉल present, and if it is an animal cell then the cell wall is absent. Now according to this cell theory, the structure, yes, the structure, basic structure of all these cells and the activities occurring inside all these cells, they are similar. That's the next point of the cell theory. Yes, so what is the next point? That all cells are basically similar in structure as well as activities. Now coming to the last point associated with the theory, जितनी भी activities एक organism के अंदर हो रही हैं, all these activities, they are the sum total of the activities occurring inside the cell. तो जितने भी फंक्शन ऑर्गेनिज्म के अंदर हो रहे हैं, दैट इज सम टोटल ऑफ द एक्टिविटीज अकरिंग इनसाइड द सेल. सो ऑन दिस बेसिस वी कैन ऑल्सो से दैट द सेल इज functional unit of all living organism. So let's write this information. All the activities occurring inside an organism is sum total of all activities occurring inside the cell. So on this basis we can easily say that the cell is functional unit of life. Right? So the different points associated with the cell theory is very much clear to all of you. Now one of the major drawbacks which was associated with this theory, that this theory, it could not explain that how the new cells they are formed. Yes. So one of the drawback associated with this theory, it could not explain how new cells they arise. Now, for the first time it was observed by Rudolf Virchow that the new cells they arise from the pre-existing cells. Yes, so here I will be writing the name of very important scientist. The name of the scientist is Rudolf Virchow. Yes, Rudolf-Virchow for the first time observed that the new cells, they arise from pre-existing one. From pre-existing cells, or he stated that the omnis cellula e cellula. Right. So after the contribution of the Rudolf-Virchow, after the finding of the Rudolf-Virchow, this cell theory was modified. Yes. So after this contribution of the Virchow, the cell theory was modified, and the cells, they arise from the pre-existing cells. This theory, this law, it is known as law of cell lineage. So this was all about the contribution of the different scientists towards the discovery of the cell and formulation of the cell theory. So now the next topic which I will be discussing here, that is the cell size. So let's discuss now cell size. Now there is great variation. There is great variation with respect to the cell size, shape, as well as the functions performed by the cell. So let's start with the cell size of bacteria. So bacterial cell size, that is ranging from 3 to 5 micrometer. So this is length of bacteria. So you might be knowing that the smallest bacteria is Mycoplasma. Yes. Mycoplasma, this represents smallest prokaryotic cell. और Mycoplasma का जो size है वो normal bacterial size से कम है. And the length of the Mycoplasma, that is 0.3 micrometer. Now the largest isolated cell is the egg cell of ostrich. Yes, so this was the smallest. Now here I am talking about the largest cell. So for the smallest I can write S here. For the largest I am writing L here. That is egg of ostrich. Now coming to the RBC. RBC, you know, they are the red blood cells. The red blood cells in the humans, they have the diameter of 7 micrometer. So this is the diameter of the red blood cells in the humans. Now one of the longest cells in the organism is the nerve cells. It is very well known to all of you. Nerve cells, they are one of the longest cells, and in the case of the humans, the nerve cells, they can be as long as 90 centimeter. They can acquire the length of 90 centimeter in the case of humans. Like the cell size, there is also great variation with respect to the cell shape. Cell size की तरह cell shape जो है वो great variation show करती है. The cells, they can be disc shape. They can be columnar, cuboidal, polygonal, thread-like, or irregular. So here I will be discussing some important points with respect to the metabolic active cells. Yes, metabolically active cells. So let's discuss some important points with respect to the metabolically active cells. It is very well known to all of you that metabolism is the sum total of all the chemical reactions which are occurring inside the cells. Yes, all the chemical reactions together constitutes the metabolism, and the metabolically active cells, they are generally smaller in size. Why they are generally smaller in size? These are due to the two reasons. This is due to the two reasons. First one, they have high nucleus to cytoplasmic ratio. This ratio is high. Yes, तो आपको मालूम है कि जो न्यूक्लियस है वो हेरिडिटरी मटेरियल है इसके अंदर, तो हेरिडिटरी मटेरियल न्यूक्लियस के अंदर प्रेजेंट है एंड द न्यूक्लियस इट एक्ट्स लाइक द ब्रेन ऑफ द सेल. तो जितनी भी मेटाबॉलिक एक्टिविटीज प्रेजेंट होती है सेल के अंदर, दे ऑल द एक्टिविटीज दे आर कंट्रोल्ड बाय द न्यूक्लियस. और अगर न्यूक्लियस बाय साइटोप्लाज्मिक रेशियो ज्यादा है तो इसका मतलब न्यूक्लियस का साइज बड़ा होगा and it can have better control over the metabolic activities. तो metabolically active cells के अंदर न्यूक्लियस का साइज बड़ा, the metabolic activities they can be better controlled by the large size nucleus. Now second one, surface area to volume ratio in these cells is high. तो सरफेस एरिया सेल का ज्यादा होगा एज कंपेयर टू दी वॉल्यूम. तो अगर सेल का सरफेस एरिया ज्यादा है, इट मीन्स द एक्सचेंज ऑफ मटेरियल विद इन द सेल एंड द एनवायरमेंट इट इज मोर. Exchange will be better, exchange will be more, exchange nutrients का ज्यादा होगा, so the metabolic activities inside the cell it will increase. So the surface area by volume ratio is high, so this will increase exchange of material in the cells and the environment. और अगर न्यूक्लियस बाय साइटोप्लाज्मिक रेशियो ज्यादा है, सो व्हाट इज इट्स सिग्निफिकेंस? द न्यूक्लियस कैन हैव बेटर कंट्रोल ओवर द मेटाबॉलिक एक्टिविटीज अकरिंग इनसाइड द सेल. So after dealing with this, now I will be discussing here different types of cells. So you know that there is two types of these cells, that is prokaryotic and eukaryotic. So I will be discussing these two types of the cells in a comparative manner.